name is Seiser. I am part of the Field Application Scientist team with QIAGEN Digital Insights. And as Deb mentioned, today we're going to do an exploration of the tumor microenvironment using Ingenuity pathway analysis. What we'll go over first is the agenda for today's session. And to start off, what I want to do is provide everybody some introduction to the major resources under the hood of QIAGEN's Ingenuity pathway analysis that really power all of the functionalities that we're going to be exploring today. And in particular, this refers to the QIAGEN knowledge base and the QIAGEN omics off Lance. So these are two major resources of information that we generate through curation efforts that allow us to do all of the functionalities in the software. Now, once we cover that, then we'll move on to kind of looking at the tumor microenvironment. And there are two primary contexts in which we're going to kind of explore the TME. One being the scenario where perhaps you start off with a target molecule of interest or start off with a disease of interest, and you want to understand how that relates to the TME. There are two mechanisms in which we're going to talk about in this context. One being taking that target or that disease and utilizing it to build networks within IPA. So there's a robust set of tools that allows you to take single molecules or sets of molecules and actually do network construction in silico. Once you build those networks, then there's the additional functionality that allows you to predict how alterations in activity of certain molecules or certain nodes within your network will influence the network as a whole. So we're going to look at that as our first topic. And then from there, we're going to transition into another aspect of more exploratory analysis in IPA. What happens when we start off with, let's say, a disease indication of interest, and we want to understand what is the immune-related set of molecules that are known to be associated with the disease. How can we define those? How can we explore those in the context of looking at public omic data that's been processed tied to those particular molecules? How can we look how those molecules might be altered in different public data sets? So that will encompass the first part of today's session. And then the second part of today's session will more focus on how we can evaluate the tumor microenvironment in the context of bringing in a more formalized expression-based data set into IPA. And there are two ways to do this. One is going through the pathway analysis section of IPA to explore what pathways are changed, in particular the TME, and how the changing molecules in that pathway might influence the activity of the pathway as a whole, how it might drive certain functional endpoints within the pathway. What this also will allow us to do is actually concurrently look at different conditions. So let's say I am doing a study where not only do I have, let's say, a disease versus normal, maybe I have other conditions, right? Treatment versus control, different types of drugs, different time points, different tissue types or cell types, right? We might have multiple comparisons. And in this context as well, working with these data sets, we can actually overlay different comparisons onto the TME pathway in particular and look at how that differentially modulates molecules within that pathway. Now, in addition to this, one other aspect that we're going to talk about with regards to data analysis with an IPA is looking at regulatory prediction within the software. So IPA has a very powerful tool that allows you to try to define what regulatory molecules might be driving the observed expression changes in your data set. And we're going to look at how we can actually use the core analysis in IPA to define a cytokine regulatory profile for a particular data set and tie that into, again, specific functions related to tumor biology. So after we cover today's topics, we'll end with the Q&A session. Again, feel free to ask questions as we go along today. There's no need to kind of hold everything to the end, and we're happy to kind of answer those as we go. So with that, let's go ahead and begin. And what I, again, first wanted to do was introduce everybody to some of the main underlying resources that power the functionalities within IPA. Those come in two main flavors, the primary one being the QIAGEN knowledge base. For those of you who are users of IPA, this is probably familiar, but for those of you who are relatively new to the software, hopefully it will inform you on what this resource is. So the knowledge base itself essentially represents a massive ontology of curated literature findings that try to define molecular interactions and associations from the literature. At its current point, the knowledge base itself has over 7 7.7 million findings that have been curated. And this resource as a whole has been built for almost 20 years at this point. And the nice thing is that it's currently and continually being updated. So every week, more curated content and relationships go into this knowledge base. Now, when we discuss the curation process itself, really this is an aspect that differentiates IPA from many of the other resources out there, because when we do curation of relationships and associations, we do so in a primarily manual fashion. So we have a team of MD and PhD level scientists whose job it is to go through the literature, find top journals, relevant recent publications, and try to define what are the key molecular interactions and associations found within those publications. And because we're going through this process of manual curation, the curators themselves understand the biology of these studies and therefore can pull out appropriate context for these relationships from the publication. And these contextual details themselves actually go into the ontology, and we'll see in a couple examples today how those can be utilized as we look at different functions within the software. In addition to our manual curation, we do use some automated approaches to highlight critical abstracts of interest, but even in that case, we have manual curation on the back end to pull out the relevant relationships. So ultimately, what this provides for you is a resource of very high quality, well understood interactions between molecules and associations between molecules and disease. Now, in addition to our curation efforts, the knowledge base also does include many third-party databases as well to supplement our curation efforts. There are a couple here listed on the slide. So again, in the context of cancer, we might be interested in things like Cosmic or clinicaltrials.gov, or maybe if you're working in microRNA, things like TargetScan, what have you. So overall, the QIAGEN knowledge base represents a rich repository of molecular interactions that are of very high quality. And another aspect that I just wanted to show everybody very briefly is how we actually go through the curation process itself. And so as our curation team goes through the literature and identifies an article of interest, here represents perhaps one study that's been identified, they will go through the relevant sections of the publication, in particular, the results of the study. So we have kind of empirical data driving these relationships. And as our curators read through these results, they'll pull out relationships, and we define these as findings. And a finding is actually listed here. So from this publication, the finding that our curators would pull out is that homozygous mutant mouse NRF2 gene caused by a knockout in the mouse decreases constitutive expression of mouse GSTM3 mRNA in the mouse liver. And so this finding essentially defines a relationship between NRF2 and GSTM3, but not only, again, do our curators define this relationship, they also pull out the contextual details of this finding. So we know that the study is done in the mouse, and in particular, it's done in the liver tissue. We know that NRF2 is the upstream molecule and GSTM3 is the downstream molecule. We know in this particular study that there's a knockout. And most importantly, and what we'll see utilized multiple times today, is when we capture these interactions, we also capture the directionality of these interactions as well. So in this context, when there is a decrease in NRF2 caused by a knockout, that leads to a decrease in expression of its target molecule GSTM3. So as we'll see today in a minute, as we go through the network construction process, all of this information essentially can be overlaid into a network. As we go through the network generation process, we can capture these relationships in a fashion such as we're looking at here, where if we look at NFE2L2 or NRF2, how it interacts with GSTM3. We first of all capture by the shapes, the type of molecules that we're working with, whether it's an enzyme or a kinase or a transcription factor, what have you. The edge itself has a terminator that will relate to the nature of the relationship. And in particular, when we talk about the directionality of effect, we know that in this study, there's a decrease in NFE2L2. So here we have that decrease represented as green. And that would lead to a prediction of a decrease of its target, as was found in the paper. So this predicted activity change of a decrease here is depicted in blue. So as you'll see today, there's lots of information actually overlaid onto the networks that we present within IPA.
QIAGEN.
Sample to insight.